Welcome back friends. Tuendelee na sasa hivi tuanze kusoma linkage au gene linkage. All the situations and examples discussed so far in this topic have dealt with the inheritance of genes situated on different chromosomes. Cytological studies have revealed that humans possess 46 chromosomes in all somatic or body cells. Since human possesses thousands of characteristics such as blood group, eye color, and the ability to secrete insulin, it follows that each chromosome must carry a large number of genes. Kwa kila chromosome lazima iwe ina gene nyingi. Now genes situated on the same chromosome are said to be linked. Zinakuwa tunazita ziko linked. All genes on a single chromosome form a linkage group. Zinatengeneza linkage group and usually pass into the same gamete and are inherited together. They pass into the same gamete and they are inherited together. As the result of this, genes belonging to the same linkage group usually do not show independent assortment. Hazionyeshi independent assortment. Kwa nini? Kwa sababu zenyewe zinakuwa ziko kwenye chromosome moja. Since these genes do not conform to Mendel's principle of inheritance, Mendel's principle of independent assortment, they failed to produce the expected 9:3:3:1 in breeding situations. Kwa katika breeding situation, kama characteristics ziko mbili hazileti dihybrid tulioizoea, involving the inheritance of two pairs of contrasted characteristics among dihybrid inheritance. Now without linkage, as we have seen, four types of gametes are produced in equal frequency. Say from the genotype ikiwa hii hapa, we get capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. With the linkage, most of the gametes will be either capital R, capital Y, or small r, small y. So tutakuwa na two gametes only, because the two loci are physically tied together on the same chromosome. So that means tutakuwa tuna let's say hapa tutakuwa tuna R Y, hapa tutakuwa na R Y. Unaona, kwa linkage ndio inakuwa inaonekana kama hivyo hapo. Examples of gene linkage. Kwanza, genes for body color and wing length in Drosophila melanogaster are linked. Body color pamoja na wing length. Gray bodied and long-winged Drosophila are dominant. Gray-bodied and long-winged Drosophila are dominant to black body and vestigial or short-winged Drosophila. Now, if pure breeding gray-bodied, long-winged Drosophila are crossed with black-bodied, vestigial-winged Drosophila, the expected F2 phenotypic ratio would be this one, ambao tuliipata kule kwenye Mendelian. Lakini this would indicate a normal case of Mendelian dihybrid inheritance with a random assortment resulting from the genes for body color and the wing length being situated on non-homologous chromosome. However, this result is not obtained. Instead, the F2 show an approximately 3:1 ratio of 3:1 ratio of parental phenotypes. This may be explained by assuming that the genes for body color and the wing length are found on the same chromosomes, that they are linked as shown below. Now let G represent the gray body ambayo ni dominant, a small g represent a black body ambayo recessive, arrow represent long wing ambayo dominant, na arrow represent vestigial wing ambayo ni recessive, vestigial wing ambayo ni recessive. So from there now, parental phenotypes takuwa na gray body long wing, black body vestigial wing. Unaona, lakini the two genes they are linked. So gray body long wing zimekuwa linked, unaona, ziko kwenye the same chromosome. Na huku a black body short wing, vestigial wing, ziko linked, unaona. Kwa katika meiosis tutapata gamete yule pale. Na hiyo ndio gametes kwenye linkage. Random fertilization manake fertilize hao wili yule na yule tutapata yule pale ambaye atakuwa ni heterozygous, a gray body long wing heterozygous, unaona. Kutoka hapo katika F2 maana yake tuta close wale wawili. Sasa F1 tuta close huyu na huyu. Katika gamete tutapata wale gametes wanne. Katika wale gametes wanne tutakuja kupata crossing, unaona yule na yule, yule na yule, tukicross tunapata yule pale gray homozygous, tunapata yule pale gray heterozygous, gray long heterozygous. Hiyo hapa ni black short. So unapata 3:1 instead ya kupata 9:3:3:1. Unaona bwana. So in practice, though this 3:1 ratio is never achieved and the four phenotypes are invariable produced, zinatengenezwa kwa ratio ambayo sio specific. This is because total linkage is rare. Yani linkage huwa haiwi total. Baadhi ya gene zinaweza zikawa linked, nyingine zikawa non-linked. Most breeding experiment involving linkage produce approximately equal number of parental phenotypes and a significant smaller number of phenotypes showing new combinations of characteristics also in equal number. Kwa kuna equal number la huku moja na huku moja. Moja ambaye ni homozygous dominant, moja ni homozygous recessive, lakini pia kuna organism ambao wanaonyesha intermediate characteristics nao wakiwa katika equal number. Now the phenotypes showing new combinations of characteristics are described as recombinants. From this, it is possible to produce the following definition of linkage. Kwa wale organism ambao wanaonyesha new combination of gene wanaitwa recombinant. Now tunaweza kutengeneza definition ya linkage. Sasa tuone. So tunasema two or more genes are said to be linked when phenotypes with new gene combinations. When phenotypes with new gene combinations or recombinants occur less frequently than the parental phenotypes. Two or more genes are said to be linked when phenotypes with new gene combinations occur less frequently than parental phenotypes. So the events leading to discovery of linkage by American Thomas H. Morgan, unaona, may be summarized in one of his experiments in which he predicted the results of a test cross between heterozygous gray-bodied long-winged Drosophila and homozygous recessive black-bodied vestigial-winged Drosophila. Sasa yeye alifanya test cross kati ya organism wawili, akapredict result. The two possible outcomes were predicted as follows. Moja, if the four alleles for gray and black body and long-winged and vestigial wing for gray and long wing, lakini pia black body, okay, for gray and black body and long and vestigial wing were different pairs of alleles. Kama zilikuwa different pairs of alleles, that is not linked, they should show independent assortment that produce the following phenotypic ratio. Unaona, independent assortment katika test cross ratio ya dihybrid inheritance tunataka tupate 1:1:1:1 kama zimefanya independent assortment katika test cross in dihybrid inheritance. But if the alleles for body color and wing length were situated in the same pair of chromosome, that's linked, the following phenotypic ratio will be produced, and that's one gray body long wing and one black body vestigial wing. One gray body long wing na one black body vestigial wing. This can be illustrated below. Kwamba tunapataje 1:1. Kwa hiyo if the four alleles were situated on different chromosome, test cross phenotypes, gray body long wing, black body vestigial wing. Kwa hiyo huku tutakuwa tuna gametes 4, 1 2 3 different, kuna tuna gametes ambazo zinafanana. So there is no need of putting all four gametes while the results will be the same. So naweka pale capital G capital L, capital G small l, small g capital L. Tunaweka huku na huku tunaweka, unaona bwana, offspring genotypes ambao ni 2n illustrated in the in each square. Unaona, one gray body long wing, gray body long wing, one huyu hapa ni gray body short wing, huyu ni black body long wing, huyu ni black body vestigial wing. But if the four alleles are situated in the same, hii ni kama they are situated in different chromosome. If the four alleles they are situated in the same pair of chromosome, test cross phenotypes, gray body long wing, black body vestigial wing. Kwa utakuja kupata hapo, tutakuja kupata hizo test cross genotypes, tutakuja kupata ile pale na ile pale. Meiosis hii na hii hapa. Haya, huku gamete ni ule tu mmoja. Hata ungeyatengeneza wawili ungekuja kupata ile ile. Utakuja kupata hii na hii hapa. Unaona? Kwa hiyo huyu hapa ni one gray body long wing, one black body vestigial wing. Unaona? Kwa hiyo ndo Bwana Morgan akasema hii inaitwa linkage. Bwana Morgan ndo event ambayo ilimfanya ajue linkage ilivyo. Note, in linked genes the phenotypic ratio in test cross is 1:1, while in non-linked genes the phenotypic ratio in test cross is 1:1:1:1. Morgan carried out this test cross several times and never obtained either of the predicted outcomes. Each time he obtained the following result. Kwa hiyo matokeo aliyopata ni 41.5 gray body long wing, gray body long wing 41.5. Unaona? 41.5 gray body long wing, 41.5 black body vestigial wing, 8.5 gray body vestigial wing, 9.5 black body long wing. Unaona? On the basis of these results, he postulated that 1. The genes were located on the chromosomes. 2. Both the genes were situated on the same chromosomes, that is, linked. 3. The alleles for each gene were on a homologous chromosome. And number 4, alleles were exchanged between homologous chromosomes during meiosis. The reappearance of recombinant alleles in 17% of the offspring was explained in terms of point number 4. This is known as crossing over, kwamba alleles were exchanged between the homologous chromosome during meiosis, ambao ni crossing over ndio ilipelekea zile gray body na black body ziweze kutokea pale. Haya, the genes for flower color and pollen shape in sweet pea are also linked. Kwa hiyo ni mfano tu mwingine wa gene for flower color and flower color and pollen shape katika sweet pea plant, they are linked. Haya, kidogo to discuss crossing over na tuone crossing over ina umuhimu gani kwenye linkage hapo. Mendel's second law is applicable only when two genes that are located on different chromosomes. Alleles or genes located at different places on the same chromosome do not separate from one as the result of crossing over. Unaona? They do not separate as the result of crossing over, and the frequency with which they separate from one another depend on the distance between them on the chromosome. Unaona? Hii ni do, sio do not. Kwamba alleles zikiwa ziko kwenye the same chromosome zita separate kwa sababu ya crossing over 2. Kwa kwenye kesi ya linkage, crossing over ni kitu cha muhimu sana ili ku-exchange genetic material. Kwa hiyo recombination between alleles on the same chromosome occur by process called crossing over as the result of physical exchange of corresponding genetic segment between two homologous chromosomes. During process 1 of meiosis, ndiko ambako crossing over inatokea. The exchange involves only 2 of the 4 homologous chromosomes. Involve only 2 of the 4 homologous chromosomes. Unaona? The exchange involves only 2 of the 4 homologous chromosomes. The alleles of parental linkage groups separate. And new associations of alleles are formed in the gamete cell, a process known as genetic recombination. Called they separate and new recombination they are formed. Offspring formed from the gametes showing a new recombination of characteristics are known as recombinants. Kwa tutakuwa na offspring ambao wanaonyesha recombination mpya ya characteristics ambao wanaitwa recombinant. Thus crossing over the major source of observable genetic variation within the population. Observable genetic variation within the population, the major source is crossing over. Na wala sio independent assortment of chromosomes, kwa sababu most of the genes they are linked and they don't undergo independent assortment of chromosomes. Test Test crossing the heterozygous gray-bodied long-wing Drosophila with homozygous double recessive flies, yani black-bodied vestigial wing Drosophila, produce the following results. Unaona parental phenotypes, gray-bodied long wings ilikuwa 965, black-bodied vestigial wing 945, recombinant phenotype black-bodied long wing 206 na gray-bodied vestigial wing 185. These results indicate that the genes for body color and wing length are linked, zinakuwa zimeshikana. Remember that a hybrid cross between an F1 heterozygous and a double homozygous recessive would produce 1:1:1:1 phenotypic ratio if genes had been situated on different chromosomes and therefore had undergone random assortment. Kwa kama zinge undergo random assortment ungepata ratio. Now consider the cross below. Consider your cross hapo chini. Test cross phenotypes gray body na yule pale naona yule heterozygous double recessive, heterozygous double recessive. Baada hapo meiosis ina show crossing over. So this meiosis ina show crossing over. So this small l itafanya crossing over, itakuja huku, small l itakuja huku. Unaona gray body long wing heterozygous, gray body long wing heterozygous. Unaona katika gamete tutakuwa na yule pale gray body long wing. I think kuna kitu kitakuwa kimekosewa hapa, lakini nichukua kwenye BS. Anyway, linkage kama linkage yenyewe hautaulizwa kwenye mtihani linkage, utaulizwa mambo ya sex linked diseases. Genetic explanation of crossing over and the reappearance of recombinant genotypes. Using the figures obtained from the cross above, it is possible to calculate the recombinant frequency of the genes for body color and wing length. Recombinant frequency is calculated as recombinant frequency is equal to number of individuals showing recombination mara 100 ganya kwa number of offspring. From the example above, the recombinant frequency or percent is tapata ni 17. This value indicates the number of crossing over which have occurred during gamete formation. So here na mark the end of our discussion kuhusu linkage. Kipindi kijacho tutaangalia sex determination na sex linked diseases. Sex linked diseases, mambo ya hemophilia, red green color blindness, na inheritance of eye color in fruit fly Drosophila ambayo tutaiangalia sana. Na tukimaliza hicho kipindi cha sex tutakuwa tunaenda moja kwa moja kwenye mutation. Thank you.